Good day everyone! Today we delve into the fascinating world of membrane lipids, phosphatidylinositol and phosphatidylglycerol. We will uncover their distinct molecular structures, explore their crucial roles in cell signaling and membrane function, and examine their emerging applications in the industry.
So before we dive into the main topic, I'll briefly explain lipids. Lipids are organic molecules mainly composed of carbon, hydrogen, and oxygen, but some also contain phosphorus or nitrogen. As you can see here, they have a head group which is hydrophilic because it contains a polar group such as phosphate that can interact with water, and two fatty acid tails. Typically, one tail is saturated while the other one is unsaturated with a double bond. These tails are hydrophobic because they repel water. Thus, its amphipathic structure allows lipids to store energy and form cell membranes. Lipids have several major classes like fats, waxes, triglycerides, and phospholipids. Phospholipids, a major class, contain a phosphate group. Within this class is a subclass called glycerophospholipids. So as you can see in the structure here, glycerophospholipids have a glycerol backbone, two fatty acid tails, and a hydrophilic phosphate-containing head. They form the lipid bilayer and play roles in membrane fluidity and signaling. Examples include phosphatidylinositol and phosphatidylglycerol, which are the models we built.
As they mentioned, structurally, PI or phosphatidylinositol is consisted of a glycerol backbone linked to two fatty acid chains, which are the oleic acid and the palmitic acid, and a phosphate group that is further connected to an inositol ring, which is a 6-carbon cyclic alcohol. Functionally, PI is best known as a signaling phospholipid. It serves as a precursor for molecules like phosphatidylinositol biphosphate, which can be hydrolyzed into IP3, and DAG, which are key secondary messengers in cell signaling. So overall, phosphatidylinositol is more than just a membrane lipid. It's a central player in cell signaling and regulation. Now, let us move on to phosphatidylglycerol, or commonly known as PG. Structurally, PG also has a glycerol backbone and two fatty acid chains, which are also oleic acid and palmitic acid, but its head group is a glycerol molecule attached via a phosphate group. Unlike the phosphatidylinositol, which have a present carboxylic acid groups, the carboxylic acid groups of fatty acids are not free in PG. They are esterified into a glycerol. That means the COOH is no longer present. Instead, you have the ester linkage of COO and R. Therefore, there are no free carboxylic double bonds in PG. PG is not primarily used in signaling. Instead, it plays a crucial role in membrane structure and stability, particularly in certain specialized membranes.
So now, let's have the biological roles. Phosphatidylinositol or PI helps with cell signaling, moving materials inside the cell, and keeping the cell's shape. Phosphatidylglycerol or PG, on the other hand, helps make cardiolipin for mitochondria, gives energy support, and helps control cell death. Together, they are essential for keeping cells alive and working properly.
The application of PI and PG extend far beyond the cell membrane, making them valuable in molecules in technology and medicine. For PI, its deep involvement in cell signaling pathways make its prime target in medicine. Researchers are using PI-related studies to develop new treatments for major diseases including cancer, diabetes, and neurological disorders. Beyond the lab bench, PI acts as an emulsifier in food industry, commonly found in chocolate and margarine, to ensure ingredients don't separate. Furthermore, in material science, PI is used to construct specialized nanocarriers for advanced drug delivery and other cutting-edge biomedical uses. Now, for PG, in medical field, PG is crucial for assessing fetal lung maturity, and it's incorporated into specific drug delivery system.
Finally, its natural amphiphilic properties make it indispensable in material science, where it functions as an effective biosurfactant or lubricant. Phosphatidylinositol and phosphatidylglycerol are not just cell lipids. They raise real ethical, environmental, and economic concerns. For PI, the ethical issues access. FDA-approved PI3K inhibitors for breast cancer cost over $12,000 a month, putting life-saving cancer treatments out of reach for many patients. Environmentally, its production creates chemical waste. Economically, PI powers billion-dollar nanomedicines like toxin. For PG, the ethical challenge is equity. PG-based surfactant therapy saves premature infants with respiratory distress, but many low-income countries lack access. Environmentally, sourcing from animals and plants raises sustainability concerns. Economically, PG drives the billion-dollar neonatal care for market, yet high cost limit its availability. Both lipids defined as medical progress has balanced ethics, environment, and economics.
Let's summarize the key differences and similarities between PI and PG. Both are vital glycerophospholipids, but they are defined by their different head groups. PI is famously known as the signaling powerhouse, crucial for cell communication. PG, on the other hand, acts as a primary structural stabilizer in membranes. Importantly, both PI and PG are essential not just for cell membranes but also have growing applications across medicine, foods, and materials.